Hi there. In this video, we're going to see how fat is mobilized, both from fat cells, which we have in storage, and from the diet. So triglycerides, so the fatty acids that we saw in the previous video, they're really, in many cases, they're found as, they're stored as triacylglycerols. So triglycerides are really esters with the hydroxy groups of glycerol. This is glycerol. So it had a hydroxy group on every carbon. It's 3 carbons, and and one or some or all of them, so could be esterified with a fatty acid, and they don't have to be the same fatty acid. It could be different fatty acids. And here is a saturated fatty acid shown at the top, a monounsaturated one with 1 double bond, and then we have a polyunsaturated one with 3 double bonds. Something else that we need to bring up here is the nomenclature. Alpha, or all the Greek letters here, they refer to the carbon position relative to the carbonyl carbon. So the one next to the carbonyl carbon is the alpha carbon, the beta, gamma, and so on and so forth. And the last one is the omega. So all the omega fatty acids you hear refer to some double bond, X number of carbons from the omega position. If it's omega-3, as I believe that's the one, or something close to it, 1, 2, 3, yes, that is an omega-3 fatty acid. It's also an omega, whatever that is, 6 and 9 fatty acid, right? So alpha and omega, they refer to carbon positions relative to the carbonyl carbon. So where do we store a lot of fat? Well, in fat tissue, and this is a typical fat cell that you see. And it's enormous, okay? And it's enormous. The rest of the tiny little things is really the nucleus of the cell. So fat is the vast majority of the size of a fat cell, okay? So fat cells really are fat, okay? That's the bottom line. What happens when you want to mobilize some of that reserve? How do you do this? Well, as I said, the fatty acids are stored as esters with glycerol, so like triacylglycerol for the most part in the adipose tissue. Then we have seen kind of like this before. I mean, the signaling cascade. So something triggers the plasma membrane on, some response on the plasma membrane of the adipose cell, of the fat cell. And it stimulates adenylyl cyclase, which we have seen already what it does, let's say epinephrine or glucagon, which as we saw, triggers also mobilization of sugar reserves in breakdown of glycogen. Well, in this case, it also causes breakdown of fat. And it's adenylyl cyclase, once the receptor is stimulated by some hormone, will generate cyclic AMP, okay, the signaling molecule, which in turn will activate PKA, protein kinase A. Right? So protein kinase will be active, and that in turn, look at the green going, the activation cascade here, will activate a lipase, another enzyme, triacylglycerol lipase, which will go on and clip, hydrolyze these esters between the fatty acid and the glycerol backbone. And it does so one at a time. So you're going to get diacylglycerol, you're going to get a monoacylglycerol, and then free glycerol. And every time you do this, you're going to release a fatty acid, which can go out. Right? Now, there is another version of lipase, not in the fat cells, but the one secreted by the pancreas, okay? And that takes care of triacylglycerols that we intake from our diet. So again, as we saw before, you're going to have the pancreatic lipase to clip one of these ester bonds, generate the free fatty acid and diacylglycerol, and then another one, you have monoacylglycerol, and then it will stop. So that's a kind of difference here. It will not release all 3 of them. It's going to leave one as a monoacylglycerol compared to the fatty acid lipase. The pancreatic lipase will leave one of the fatty acids esterified with glycerol. So this is a view of your intestine as it comes in from the stomach, and there is a gallbladder, and what you get is the fat droplets in the diet. They come in food, and then the gallbladder and all these juices flow in, and you have and from the pancreas, and you have bile salts and other things, and then you emulsify these triglycerides so they can actually float in solution there. When the lipase works on them, you get the two free fatty acids from each triglyceride, and then you have a mono glyceride. Okay? Those get then put together in micelles, shown here. So you have bile salts and the micelles, and they all come together, and that's how they can be absorbed, okay, by the intestine. So these are the brass cells in the intestine, and they take the fat part of the micelle, the bile salts stay behind, okay, and then they put them together as triglycerides and protein in whatever is called a chylomicron. So you have a protein surrounding the triglyceride, and they come out, and then they get into the circulation as chylomicrons. So what happens to glycerol after triglyceride breakdown by the lipases, as we've seen? Well, that actually we know what happens. It can enter glycolysis. It can be utilized, the carbonoglycerol. It's not a sugar yet, as I said, glycerol. It has to be made. One of these hydroxy groups has to be oxidized to a carbonyl, and to become, in this case, the hydroxyacetone phosphate. It's carbon 2, and before that, the glycerol is phosphorylated at position 3 and becomes a glycerol 3-phosphate, and then you have the carbon oxidation. You get dihydroxyacetone phosphate, as we have seen, and that's how glycerol gets utilized. So last thing about fatty acids and how they can be found in the serum. So we have a very abundant protein, all animals do, all mammals do, albumin in the serum, and I mean that's the structure, and it takes about more than half, at least, of all the protein content in the serum. So it's a huge amount, and it binds just because of its presence in such high amounts. It can bind and transport a variety of things, drugs, hormones, and also fatty acids, as it turns out. So serum albumin is very important. It maintains osmotic balance and the pH of our serum or blood. Another thing that we need to remember is also what other downstream effects that are caused when we take, let's say, that when we take a lot of dietary fat. So it turns out that's a signal transduction pathway that triggers gene expression. And there are specific receptors that bind this dietary fatty acid triglycerides from the diet. They may come in, okay, and then they bind to these so-called receptors that are found in the cytoplasm. But upon binding with a fat, they translocate in the nucleus. This is the PPAR receptors, as they're called, and they go in the presence of fat. They go into the nucleus and they trigger gene expression. And that gene expression has very important outputs, which is it increases inflammation on one hand, but also it increases fatty acid metabolism. So you actually break down fat. If this system, if it senses a lot of fat, it triggers the necessary patterns of gene expression to stimulate fatty acid breakdown, so you can get rid of it. It's actually quite effective if it gets stimulated, and in fact, current drugs that are used to lower triglyceride levels, for example, in the blood, these hypolipidemic drugs, they are like Fibrozil, Lopid, are very popular. I mean, many people are on them. Millions and millions around the world. They work exactly the same way. They bind these PPAR receptors and they trigger this response so that fat can be cleared from the circulation. All right, so fine, we got the fat. In the next videos, we're going to see how we actually break it down.